Hello, everyone, and welcome to another podcast broadcast of This Week in Science. Justin and I are back again. Yay! Let's talk about science. We're going to talk about other things. We'll announce something in just a minute on the podcast, but it's not the podcast yet because this stuff gets edited out because this is pre-podcast, and then there's post-podcast, and then there's other mess-ups that sometimes get edited out by the wonderful Rachel. And so if you're watching right now, thank you so much for watching. Everyone in the chat, thank you for chatting. We love you being here. Okay, we're going to do this show. Not everything will show up in the podcast. Please press the likes, the shares, and all the things that let other people know who don't already know that this show is happening right now. Yeah, I know. All right. Do you go like that? Is that what we do? Is that the...
I don't know.
I don't know.
You say, oh, there's a button.
Mash that
like
imaginary button of like. Okay, we're going to do a show now. You ready? Justin looks super ready. All right.
Hey, wait. Right here.
What?
Here,
no, I'm going to say it. What?
I know you're going to say the thing, but for some reason, when we go to single shots now, it does a extra zoom. So it's not just the shot that I see here. Mm-hmm. Right here, view is perfectly framed. When it goes to the single shot, it zooms in, and you lose. Yeah, I don't know what that is. That's kind of a new. I've seen it last like a couple of weeks. Anyway,
this is a new. We use StreamYard for our broadcasts, so it's their system. I can reframe myself so that doesn't happen in the future. But
okay.
Thank you for bringing it up.
Yeah.
Yeah. Let's go foreheads and science. Starting the show in 3, 2. This is TWIS. This Week in Science, episode number 939, recorded on Wednesday, August 16th, 2023. All in all, we are all just bricks in the science. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with music, flies, and extinction. But first...
Disclaimer. In Mesopotamia, around 5,000 years ago, the Babylonians were recording astronomical observations, laying the foundation for the scientific study of the cosmos. Ancient Greeks invented the computer 2,200 years ago, the Antikythera mechanism, a masterpiece of advanced mathematics, astronomy, metallurgy, and engineering that could track celestial objects, predict moon phases and eclipses, and still get you to the next Olympic event on time. Pythagoras and followers explored the mathematical relationships governing the universe. Aristotle brought a systematic approach to scientific inquiry. The Islamic Golden Age brought a comprehensive medical encyclopedia that influenced medical surgery practices for centuries. Algebra was invented. Chemistry and astronomy advanced. The Renaissance man, Leonardo da Vinci's anatomical drawings and technical designs and equations of gravity. Nicholas Copernicus and Giordano Bruno challenged the Earth-centric model of the universe. Galileo Galilei's telescopic observations confirmed them. Isaac Newton's Principia Mathematica brought together the laws of motion and universal gravitation, providing a unifying framework for understanding the physical world. Charles Darwin's theory of evolution by natural selection reshaped the understanding of life's diversity and origins. Albert Einstein's theories of relativity upended classical notions of space, time, and gravity. The Copenhagen interpretation, led by Niels Bohr, challenged our understanding of the subatomic world, and the discovery of DNA structure by Rosalind Franklin revealed the inner workings of all biological life. And now, just now, it's being announced that there is another episode of This Week in Science coming up next.
I've got telepathic kind of mind that can't get enough. I wanna learn everything, I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Justin, and everyone out there. Blair is out this week, and we would love to send her and her partner, Brian, an immense congratulations because Blair has brought another human being into this world, and who better to do so?
Really wild.
Right? Wonderful, caring people, intelligent, curious. I'm so excited, and I hope everything's going well, and I hope that all of you out there use whatever social medias or whatevers and, do the yay, congratulations, give them the love, because they have now embarked on a whole new adventure in life.
Yes,
Bringing life.
One that does not include sleep.
Yes, not a lot for a long time. We'll see how that one goes. But thank you, everyone, for joining us for another episode of This Week in Science. We have, Justin and I, lots of science to share with you. I have stories about dead flies, sexy flies, fake meat, and demons.
Whoa.
What'd you bring, Justin?
I've got music from brainwaves, hidden consciousness in coma patients, the extinction of all humans in Europe, and the best darn dino tracks anywhere in the world.
Dino tracks like music, dino, dino.
Yeah, it's a teaser. It's a teaser. You have to wait around and find out. It's going to be at the end of the show. So
looking
just fast forward
to
if you're on the podcast or YouTube. You can just find out real quick and then come back.
And in the meantime, I'm going to tell everybody that if you are not yet subscribed to This Week in Science, you can do that. Look for This Week in Science on all the podcasting platforms that you're familiar with. TWIS is another name we go by. We're also on some social medias as TWIS Science, TWIS CIENCE. We broadcast live weekly on YouTube, Facebook, and Twitch. Look for twis.org in the interwebs, and there's all sorts of our website with other disinformation, and you can get show notes and links to stories that we talk about.
I always feel like we're giving directions to a party that somebody's already at.
Yeah, a little bit, but maybe there's that many.
Like if you're listening to this, you already found us. They're already at the party. You don't have to. Whether you're going to take a left before you get to the big.
Okay, here's the thing. This is the party where you're allowed to invite your friends.
Oh, okay.
So tell your friends.
That makes sense.
Yeah, it does. You ready for some science?
Yeah, let's bring it.
Yay! It's a demon-haunted world for real.
This show is already just. What are you talking about, Kiki?
I know. What am I talking about? Okay. 1956, there was a theoretical physicist named David Pines, and he predicted that there are demons in matter.
Is this like little animals that follow you around and share your thoughts or something? What was that?
Nope.
Golden compass situation?
Nope. This is not. I don't think so. Nothing to do with that. But we know that in materials, especially those that form structures where electrons like to combine and act in a similar way, and they're like, we're happy now, we're sad now, we're happy now, we're sad now, and everybody, the electrons are like, we're doing it all together. And so that's like a nice frequency of the matter and how the matter is oscillating in space and time. Well, this guy, David Pines, he was like, not all matter seems like it would do that. Like maybe sometimes there's like a couple of different frequencies, and maybe they're kind of opposite each other. And so researchers in the last several years definitely showed that there are these things called plasmons, where the electrons combine and have this wonderful massful, great interaction. And this is good for superconductivity. It's good for all sorts of stuff. But David Pine's idea was like, well, maybe sometimes they combine. Because of the way they combine, they're like the opposite of those like mass-full electrons. Maybe they're massless, and maybe they don't interact with light, and maybe, just maybe, they're like these weird things hiding in matter that we can't detect. Let's call them demons.
Okay.
So it was predicted very long time ago. And researchers were like, oh, that's a great idea. Nobody's been able to figure it out and see it prior to now. So there is a new study that is just out of University of Illinois, Urbana-Champaign, and researchers kind of accidentally found these demons. They were looking at samples of a metal called strontium ruthenate. And it's kind of similar to the metals that are used that work as high temperature superconductors, except it's not. So the researchers are like, what's going on with this metal? Why is it acting the way that it is? What are its properties? What's happening there? So they started trying to look at the electrons. They're trying to see what's happening within this substance. And they found that there were two different frequencies, and they were kind of like parallel to each other. And there was an electronic mode that had no mass. The researchers say, at first we had no idea what it was. Demons are not in the mainstream. The possibility came up, and we basically laughed it off. But then we started ruling things out. We started to suspect that we had really found the demon. The researchers calculated the features of strontium ruthenate's electronic structure, and they performed all sorts of microscopic calculations, and they found that it was just as Pines had predicted, that there was a demon observed as a 3D acoustic plasmon in strontium ruthenate, published in Nature this last week. And it wasn't what they were trying to do, but they saw it, and like they said, they tried to laugh it off. They couldn't.
So I can't tell if I'd be like really impressed and excited to find out that my experiment had discovered the demons, a 70-year-old paper confirming it. Or if I'd be like, somebody already figured this out. Like 70 years ago, somebody already wrote the paper on it.
No,
it's not like, as big a discovery because somebody... But what's going on in the 1950s that people are figuring out the entire future of hidden physics?
It was the future
on paper. They didn't have the equipment, so they're like, I'll just sketch everything out with math and figure it out. Oh, look, what I'm discovering with math on a piece of paper. And then 70 years later, a physical experiment will be able to find a Higgs boson or this demon state of electric matter. Like, that's wild.
It is wild. And it goes along with, we need theoretical physics to do the math and see what's possible, predict lots of different things. And then we get the technology and we do the experiments and we do the measurements. And sometimes the measurements don't make sense. And so we get rid of other possibilities until it's the only possibility is the reality of a prediction that's 70 years old.
Yeah. So that's because there wasn't like that many papers 70 years ago.
There were a lot, but not as many.
No, there weren't that many. Like we have I think something 5,000 papers come out a day or something ridiculous now. Then it was like maybe that many in a year. Yeah. Like it wasn't like people were just cranking out theories. You came up with one in your career and you wrote it up and you presented it. That's my idea.
But I do
have to come up with papers all the time.
No, yeah, the publisher parish is a bit of a trap, but I do love that they went into this trying to go, okay, there's this metal that we know of that is like other superconductors, high temperature superconductors, but it's not high temperature superconducting. So why is it different? And then they made measurements that came out with this result that they did not expect at all. And that in itself is, very exciting and pretty cool. Electrons. We got a lot of learning to do. A lot of learning. Want to tell me a story? Sing me a song?
Yeah. So it's just a snippet of a song.
Just a snippet, but we're not going to play the snippet.
We are going to play this. We're going to play the second snippet, which is, so this is neuroscientists at Albany Medical Center, which I think is Berkeley, recorded activity from electrodes inserted into the brains of people with epilepsy. The doctors had placed 2,668 electrodes into the brain of each of 29 patients to record brain activity during a seizure. Having gotten the folks completely wired up, they decided, hey, while we're here, let's investigate how the brain responds to music. Part of the brain responsible for understanding spoken language tends to reside in the brain's left hemisphere. But there have been studies that have suggested, well, when you're listening to music, it's not centered in the language deciphering part of the brain. It's more complex. There's a network of brain regions involved in appreciating music, which might even include both hemispheres of the brain. So subjects listened to various snippets of songs while their brain waves were being recorded. Team used these recordings to train a computational model to recognize which patterns of brain activity corresponded to which musical features. So do you have the snippet of the brain wave?
I do have the snippet of the brainwave, and we'll play it right now. Let's see if people can recognize it.
Can't hear anything. You're muted too.
I keep going back and forth with all the buttons. Okay, hold on.
So I'm not gonna. I know what the song is. I'm not gonna say it. What the snippet is. But just listen to it first.
Yeah.
Yeah. Oh, but don't look at the screen.
Here it comes.
If they look at the screen, it's. Yeah.
Should I play it again?
Time? Yeah. So now. Now, if you haven't gotten it, I will give, if you're listening along to this, not looking because it's saying the name of the thing up there. If you're listening along to this, I'll give you a hint if you haven't figured it out. It's a intro to a Pink Floyd song. Can we play it one more time? Intro to a Pink Floyd song if you're just listening along. All in all, it's just another brick in the wall. That's from Brainwaves.
All in all, from
Brainwaves. Now I've told you this amazing thing that they can read your brain using. I'm going to take it away. So
you're going to take it away. Okay.
So here's the part that I don't understand and would want to talk to them about a little bit. So this model was able to reconstruct recognizable version of the original song. Some of the electrodes function somewhat like, they say, like a single piano keys representing individual notes. The algorithm also needed to interpret things like some acoustical elements, volume, rhythm, that sort of thing. But then it says specifically the researchers trained the algorithm on about 90% of the song. Well, wait a second. If you've trained it on the song, then does the algorithm already know what the song is before it interprets it? Because then is it just matching it? That's the part I don't understand.
But it has to start somewhere.
Yeah. Still.
Maybe it's matching and pattern recognition, but it has to start somewhere.
It has to start somewhere. You have to be able to connect those brainwaves to what's being heard. But if it's just trained it on, okay, this should sound like this should look like this, if you hear this sound like that, fine. And then, by monitoring the brainwaves, they can actually hear the song. So the researchers hope that this will be used in the future for mind reading. Oh, wait. No. The team said they weren't interested so much in the... they're interested in the mind reading aspect of this, but they're more interested in identifying the brain areas responsible for perceiving different musical features. And part of this is because they might be able to one day use it with patients who struggle to speak as a result of strokes, injuries, degenerative diseases, and allow their minds to communicate directly.
The mind's communicating directly. That's like the big one. No. It would be amazing to have technology that could enable people who are in a locked in syndrome to communicate, right? To talk with people. There's so much as to the potential of this. We've already looked into the visual cortex. We've done some speech recognition, rebuilding of speech in some experiments, but this with music is a different way of looking at it, and it's pretty interesting.
Yeah, and we would play you the original version of this. Somebody's asking, is that the choir? No, that was Waters. It's not Brick in the Wall Part 2. It's Brick in the Wall Part 1. I guess I should have said that at some point. Because it's
for the aficionados out there, you got to know.
Yeah. Anyway, we're afraid of the YouTube algorithms. Like they might be like, oh, that was close enough, and we get booted over everything here on this little niche show on the YouTubes. So for the YouTube portion of it, at least, we're not going to play it.
I think the other aspect of this, though, aside from the music, whatever, it's, these were epilepsy patients. This was not outside the skull. These were electrodes placed on the brain itself within the skull. These were people who were kind of waiting to have their surgeries done and had holes in their heads. And so it was like, oh, we're already in there. Let's do this. They're on the surface of the brain. And in that, we're still a distance away from the resolution that would be necessary to have a little cap on your head that could read these, you listening to music or imagining music in your head. There's a ways to go still, a long ways to go.
Yeah. And by the way, they specifically chose Pink Floyd for a number of the snippets that they use in this because it scientifically had been determined to be everybody's favorite band. Whether they'd heard Floyd before or not, they were like, this is just good music.
It's just good music. So, whatever. It's all good. The people's auditory cortex likes this stuff. The other side of it that I really liked is we think of where language is located, and we've talked about where in the brain music should be. And while it's a question as to where the lyrics are interpreted and processed and listened to, the researchers also point out that their results show that music is more cross hemispheres, both sides of the brain, but more right brain. More right brain oriented than left brain. Language is more left brain. So it's that in itself is interesting.
Yeah, in some people.
In some people.
Most people. And most people.
Most people love flies, don't they?
No, nobody likes flies.
Everybody
Except
loves
other flies.
flies. Flies are amazing. Right? Are they awesome? Flies. Well, some researchers presenting at the fall meeting of the American Chemical Society are presenting about their work trying to make plastic out of black soldier flies. These nice, just little flies.
Okay. Okay.
Just
Wait,
flies.
plastic?
Plastic. Yes.
Would it still be biodegradable, or would it be just unbiodegradable fly plastic?
Degradable, digestible. In fact, they do want to create a circular system in which the plastics that are created from the chitin powder that is created from the exoskeleton of these dead black soldier flies is then later fed to flies so they can live. The waste products are fed to the flies, and then they can. So they want to create a circular fly economy.
Oh, that's dark, but okay.
It is. So researchers previously have looked at glucose that comes from sugarcane, from trees, from all sorts of products that also have other uses. And so economically, if you can come up with a source of glycosides, glucose-based polymer products, you have the potential to not have like weird pricing in the market and have it's just its own thing, right? And so you can harvest the resources just from the flies, and that's great. The flies are great. So these researchers, they have been trying to work on these natural products harvested from regular resources for a while. Started working with another researcher who's trying to work on expanding the black soldier fly industry. And so they worked on examining these dead flies, found that the chitin could be used to make a sugar-based polymer. And manufacturers already use chitin from insects, shrimp, crabs, things that are kind of waste byproducts, but we don't necessarily need to use shrimp and crabs for these things. We like them for food. Anyway, can we use insect carcasses? Make plastics that don't cause seafood allergy triggering? Could. Fly larvae?
It's still very dark, but I mean, I guess that would be, in the realm of life. It's just the feeding it back to the flies thing. I think that's the
part. But yeah, it does it for me as well. But I think they're not thinking that 100% through, because there's always energy loss at every trophic level.
Of course,
so there's. But
it would have to be supplemented,
of course.
Baby grasshoppers or something.
They're hoping they can also use these chitin powders to create, use the polymer to create stuff like chitosan that can actually be used to create super absorbent hydrogels that could then be put into the soil to allow soil to hold more water, especially in drought areas. So the soil would hold more water, and then it would be better for agriculture and all sorts of things. And it would degrade and not cause a lot of microplastics problem. It's the fly science.
Yeah,
flies. Yeah. Flies. Who needs them? Maybe we do. It's possible. Okay, tell me another story.
So I have for many years had a standing demand, not a request, an ultimatum of the highest kind possible, not to be unplugged if I fall into a comatose state of outwardly unresponsive brain activity. I assume that somewhere in the recesses of my brain, I will still be functioning, having a perfectly enjoyable conversation with myself, and I never needed any of you anyway. Actually, I did, but I've done enough reading that I've got plenty inside to work over for a very long time. Now, a study by Columbia researchers has finally offered a rational perspective to my request. Demand, non-negotiable. Researchers have identified brain injuries that may underlie hidden consciousness, a phenomenon in which brain-injured patients are able to respond to simple commands, making them appear unconscious, or unable, to respond to simple commands, making them appear unconscious despite having some levels of awareness. This is quoting the study leader, Jan Closen, Associate Professor of Neurology at Columbia University. Our study suggests that patients with hidden consciousness can hear and comprehend verbal commands, but they cannot carry out those commands because of injuries in brain circuits that relay instructions from the brain to the muscles. Findings could help physicians more quickly identify brain-injured patients who might have hidden consciousness and better predict which patients are likely then to recover from rehabilitation therapies. So hidden consciousness, this is also known as cognitive motor dissociation, occurs in about 15 to 25% of patients with brain injuries stemming from head trauma, brain hemorrhage, or cardiac arrest. That seems high. In previous research, Closen and colleagues found that subtle brain waves detectable with EEG are the strongest predictor of hidden consciousness and eventually recovery for unresponsive brain-injured patients. But the precise pathways in the brain that become disrupted by this condition were unknown. In the new study, the researchers used EEG to examine 107 brain injury patients. The technique was able to determine when patients are mentally trying, just unable to respond to commands such as keep opening your hand or tighten your hand. The analysis detected CMD in 21 of the 107 patients. Researchers then analyzed structural MRI scans from all the patients looking for patterns. Researchers found that all of the CMD patients had intact brain structures related to arousal and command comprehension, supporting the notion that these patients were hearing and understanding the commands, but were simply unable to carry them out. This is a quote from Clausen. We saw that all the CMD patients had deficits in brain regions responsible for integrating comprehended motor commands with motor output, preventing CMD patients from acting on verbal commands. Findings may allow researchers to better understand which brain injury patients have the CMD and, of course, how to treat it. And more importantly, they provide a scientific basis for keeping Justin plugged in. Now, I do want to point out.
Yes.
I do want to point out.
Please do.
That this also has to do with the awareness of the outside world still being persistent. I still don't need that.
You're fine.
So if I fail this test, if it doesn't turn out I got the CMD, still do not unplug. I don't need you, any of you. I did early on, and then sometime in the mid-20s, I'd read enough stuff that I'm like, I have enough to work with now. The rest of this is just
composing novels within your brain. It's
composing novels, solving mysteries of the universe, whatever it is. I'm fine.
I think the great thing about this is that it starts to get at a methodology that is more achievable by more medical institutions. So things like MRI, fMRI, more complicated imaging techniques, they're not available to every hospital. And so if you have a technique that you can work out that's like, hey, here's a brain signal you can get by EEG, which is, a little more available to medical doctors. You look at it, you go, oh, we got the signal. Maybe we need to transport this person to a facility that can confirm with MRI. And so you start really getting at the root of being able to treat patients correctly which has been a huge question for a very long time because we haven't known. We don't know.
Yeah. Yeah, like before it was like people were in a coma and you decide, well, pull the plug, don't pull the plug. Some people just they're not plugged in. You plug you just now their
just
phone
there.
isn't charging.
Yeah,
that's all. But then people come back.
Mm-hmm.
People have come back from these states and just become functional again at some point. And it's such a mystery is what was preventing. Of course, it's networks in the brain being able to communicate and the plasticity of the brain being able to repair itself at some point, then re-establishes those controls in some.
But the being able to detect these very subtle, nuanced brainwaves, this is really a step forward. So this is very
hidden consciousness.
It is.
I sometimes feel like I have hidden consciousness already, even like in my conscious state. Like there's another hidden consciousness that's thinking about something else.
Yeah, that's a whole other thing, psychological thing. That's not the same as what you're talking about.
That's something I should take up with my therapist. Is this what you're saying?
Yeah, maybe. Okay. No, or just don't. Whatever. Just leave it. Push it deep down inside. It's all good.
That's usually how I like, stay hidden. Don't let them find out.
Never let them know. They'll think you're too weird. Oh my gosh. What? We're all weird. That's the whole thing. What do you think about fake meat?
So, in a few cases, I'd have to get brand specific and item specific. You
don't need to do that. Yeah.
I've been like, wow, this fake chicken frozen patty thing is actually darn good. I don't know why I'd bother going and get a chicken one versus this. This is perfectly acceptable. And then I've, one of those hamburgers were like, hey, it's a hamburger, but it's made out of things, but you'll never know. And the first bite, you're like, oh no, this is different. and even if you called it not meat, I might have been like, oh, this is a different thing to eat. But you called it meat, and so I took a bite and expected that, and it was different. If you just called it like a fungus burger, I might have been like, well, I'll try that. And I go, oh, this is what a fungus burger tastes like. Oh, that's interesting. Yeah, that's not bad at all. But you called it meat, and so I took a bite, and it wasn't meat. And now I'm like, even if it was fine. So, yeah. I think they set themselves up for disappointment if they call it meat, and it's really not there.
I think so. So some researchers just 2 stories out this week actually related to different kinds of fake meat. A research team just publishing in Nature Communications has been trying to make fake meat juicier, more palatable.
Put that hema in there. Put that blood, that fake blood.
But not blood, but it's the fat, right? So when you really like a burger, if you're a meat eater, a lot of times it's the fat, it's the marbling, it's the, it's that juiciness that comes through that makes you really appreciate this thing that you're chewing on, as opposed to the very dry, powdery thing that was the fungus burger that you tried to eat just a moment ago, but are now like spraying as you talk. These researchers, in trying to figure out how to make the meat more palatable and better, said, what if we just add water? What if we just made like juicy is juicy? Add more water. And so they created a hydrogel plant protein, basically a lattice that can be part of the plant protein that is whatever the fake meat is going to be. And the microgel goes, yay, I like water. I like the plant proteins. I like all of it. And it pulls it all together and makes an ultra lubricated fake meat for your mouth hole. In this, they say they offer a unique platform to design the next generation of healthy and palatable and sustainable foods. They used atomic force microscopy to figure out whether or not they're molecules were doing what they wanted to do, and the researchers say seeing the images from the atomic force microscope was an exciting moment. The visualizations revealed that the protein microgels were pretty much spherical and not aggregating or clumping together, and so we could space out the plant proteins within it. And our theoretical studies said this is what would happen, but it's nothing like seeing it for real. And now the researchers are wondering if not only they can add this to the future of fake meat, but also to reduce fat in foods that we currently eat that are processed that have fat added to make them more palatable. So can we use this microgel to make things juicier?
Hey, what would happen if we added it to ground beef to make a hamburger with? Would it make it even better?
What if you took a 1% fat ground beef thing and added this as opposed to like the regular fat ground beef? I don't know. I'm not in charge, but this microgelation process does have potential, and we'll see where it goes. It's not adding anything odd to the food. It's adding water, and that could be better for a less meaty future. Additionally, a different group of researchers decided that they're going to try and figure out how to make great fake seafood. And that's a problem. So this is, again, they're going to present the results at the fall meeting of the American Chemical Society. And we all know seafood, it's not fished sustainably very well. We have issues. Who knows how long seafood's going to last? When are we going to be eating the jellyfish? I don't know. But these researchers decided that we're going to approach this. And so calamari, let's do it. Make some deep fried calamari rings, 3D printed from a microalgae protein and mung bean protein. And mung bean protein is also a byproduct of some mung bean processing and other things. So it would be using stuff left over from the use of the mung beans. So potentially we'll be able to have these mock seafood products in the future. They think that this microalgae will work in a non-immune stimulating way. We wonder sometimes some algae supplements and some algae based products have caused allergies. But we hope that these will not, and they don't think that they're going to specifically. But the question is the future? Is this the
soldier flies?
We could use some black soldier flies, exactly. But is this the beginning of like the replicator? Is this the beginning of Star Trek and the replicator? I would like calamari. And you wait for it to be produced.
Yeah, I have never been impressed with calamari to the point where the fungal black soldier fly combo version of it would be just as palatable.
Probably. It depends. Some people love calamari, other people don't. This could be 3D printed to be other products that are seafood related. They've given it a nice fishy flavor. It's got lots of omega-3s, very healthy. 3D printing. And do I find it fishy? I didn't make that joke, although obviously I did. This is This Week in Science. Thank you so much for joining us for this new weekly venture into the world of science. We do appreciate you being here with us once again as we, Justin and I, this week, discuss the stories that we think are most interesting to bring to talk about with you. If you love the show, please head over to twis.org, click on the Patreon link, and become part of our supporter community on Patreon. You can choose your level of support, $10 and more per month, and we'll thank you by name at the end of the show. And if you can't do that, please just tell a friend about TWIS. We really can't do this without you. Thank you for your support. All right, time to come on back now, except Justin just disappeared. He honestly just disappeared. So I think he was playing with a cord. And I don't know. You want to see? You want to see? He went bye-bye. I don't know where he went. He's gone. He played with the cord and he disappeared. I'm going to wait by my phone and see if I need to make any arrangements. I'm not seeing any text messages. Would anyone like to join me for a plate of fried 3D microalgae mung bean calamari rings? Maybe, possibly. We only have a few stories to go, but I don't know where Justin went. He decided he was going to leave a little early, apparently, even though he has some wonderful things to talk about. Oh, there he is. He's back now. Oh, nope. He gave me the 1 minute signal. He'll be right back as he figures things out. We're going to just keep on talking here right now. And potentially, Rachel's going to be editing all of this out. That's the way we do it.
Yeah, so, yeah, it's the
lighting
new,
and pluggings. And, yeah, new normal.
Yeah. So, let's see. What do we got?
But hold on 1 second. Let me just say, we're all going to come back right now for more This Week in Science. Justin, do you have some stories for us? This is editing for Rachel. Live from Videoland. It's technical problems. I see plugs. And we're hanging out. There's light coming. But I agree with Justin. I don't want to eat a fake burger. I just want to eat.
I know you don't agree with me. That wasn't my point.
No, I don't.
I would totally love a fake burger. Just call it a vegetarian burger.
They do that already.
Burger.
They do that already. But
yeah, and that's
fake
fine.
Meat, it's not.
It's when you call it fake meat. That's when it becomes a problem.
Even better than the real thing.
Anyway.
Even better than fake blood. Even better than the real thing. Are you lit up?
Yeah, I'm trying
Oh.
Okay, so where were we? We were talking about science.
Justin, you got some stories for us?
Yeah, once upon a time in Europe, over 1 million years ago, there were humans. Not current modern humans, other humans, before the Neanderthals. There is a kind of poor record of these humans. Some debate and controversy about how to define them within the hominid family tree, and how they may have played a role in Neanderthal, or possibly even modern human evolution. But now, perhaps, much of those considerations can be laid to rest. Paleoclimate evidence has revealed that around 1.1 million years ago, southern European climate cooled dramatically and likely caused the extinction of any archaic humans on the continent, according to a new study led by the University College London researchers.
Ouch.
So there's this Homo antecessor. It was like, oh, this one is the precursor to Neanderthals. It's already in Europe. It's probably the wrong, but it was probably the wrong for a lot of reasons. As the just ancestor, there could be a version of early hominin that did through a different tract, through a related ancestor, become a Neanderthal. The problem was the anthropologists in Europe were trying to make a progression of everything that they saw linearly through evolution. And as we've been learning over and over and over again, that's not how hominin evolution necessarily works. You don't go from one form to another. You have lots of disparate forms, and then they recombine, and then they become other disparate forms, and then they recombine. It's the braided stream. It's all of this. That whole linear evolution thing isn't how isn't how humans have sort of gotten where we are. Anyway, this is published in the journal Science. The team of researchers discovered the occurrence of this previously unknown extreme cold snap around 1.1 million years ago. The glacial cooling pushed the European climate to levels beyond what archaic humans could tolerate. So this is the oldest known humans remains in Europe are 1.4 million years ago. Thought to have arrived from Southwest Asia, which is also, they think it arrived from there because there's other things that seem similar to it in Southwest Asia. But then again, braided stream, all of these things will be updated at some point. Doesn't matter. The climate around that time would have been generally warm and wet when these early humans got there. So they might have had some mild cold periods, but nothing severe. Up till now, the prevailing theory is that once these humans arrived, they were able to survive through multiple climate cycles and adapt to the increasingly harsh conditions. But this is senior author and professor Kronos, it's a Greek name, oh gosh, Tidakis, UCL Geography, said, our discovery of an extreme glacial cooling event around 1.1 million years ago challenges the idea of continuous early human occupation of Europe. So they did some modeling. This was actually all based on marine microorganisms. And then they went further and examined pollen content in deep sea sediment cores to reveal, these were all off the coast of Portugal, to reveal the presence of an abrupt climate change that culminated in extreme glacial cooling. Ocean surface temperatures off Lisbon dropped below 6 degrees, and there were semi-deserts expanding on the adjacent lands. Lead author Valsik Margari, also UCL Geography. To our surprise, we found that this cooling was comparable to some of the most severe events of recent ice ages. They ran some modeling, and they found that there's basically no way that small bands of hunter-gatherers could have survived, especially since early humans may have lacked adaptations, sufficient fat insulation, also the means to make fire, effective clothing, or other shelters. So that's the thing, is I can't even imagine, you've got this looming climate crisis, no way to prepare for it. And that would just be tragic.
You know nothing. You know nothing. You have no idea it's coming. It's just coming.
Yeah.
Because it's the planet. It's not you, it's the planet.
So they also kind of find this backed up with a somewhat of a mystery in the archaeological record, where there's some 200,000 years where there's no stone tools or remains being deposited. So, looks like humans got wiped out, which and then other humans started moving in after this period. This is where Justin should pivot to Justin doesn't believe Denisovans were Neanderthal cousins, but the
your Homo
hybridization
erectus theory, right?
Yeah, but it does make sense that the Neanderthals become such a distinct hominin for a really long period of time in one region. It makes more sense if the Homo erectus, which has spread throughout all of Eurasia and Oceania, just isn't there. It makes a lot of sense why they would be sort of distinctly, isolatedly classic Neanderthal for such a long time. Whereas
death.
Because there was
death.
Because they had the open human hominin niche there that they could fill, compared to the Neanderthals that went that headed east, that became Denisovan, and I think likely interacted with late Homo erectus. Although late Homo erectus, also the further you go along late Homo erectus, like everything technically, I guess at some point be late Homo erectus. Like they have like the Homo floresiensis, the little hobbit, One of the things they think, oh, that's a late Homo erectus, that island dwarf isn't. And then there's the other one, Luzensis or something else. It's very much Hobbit-like, but instead of Flores, it's over in
a
the
different
Philippines.
island. Yeah.
It's in the Philippines. It's another one. Also, the one in the Philippines, the indigenous people have the highest rate of Denisovan DNA out of anybody, at around 5%.
Which is very interesting. But I think what's really fascinating here is this just kind of adds another, I don't know, is it another thread to the braided stream or another brick in the archaeological history wall of humanity? To bring it back around.
Yeah. I don't know. So
what else you have?
If you have, I have 5-6% Native American DNA or ancestry.
Ancestry.
Yeah. From the DNA, right? From a 23andMe thing. That means, and that fits, because I have a great grandmother who was Native American. When you're talking about 5% in a population that's existing today, that doesn't mean that everybody's great grandmother was a Denisovan, right? In the larger population, when you're attributing something to an archaic, you're not talking about ancestry. It's a little different. I just want to clarify that.
Yeah, that's an important
what you're talking about
clarification.
is conserved DNA. And what's likely being conserved is also selective. And what's really curious is there's been a lot of information about genetic responses to things like COVID, long COVID, severity of COVID, based on genes. And I think it was the Neanderthal genes that they found that were like, oh, this reduces your chances of getting the infection by 23%. However, apparently whatever enzymes eating up the virus in that phase of your immune response, there's another immune response that apparently was selected for a different type of viral infection, because you're more likely to die from COVID if you have that. So it's like a double-edged thing. But it's likely some sort of immune or survival-related gene selection for that much to be conserved for that long. So there might be, I think you were talking about this a couple weeks ago. There may be things that medical science needs to look at. Therapeutics and the genetics of whatever. Why are these conserved? What are they doing?
Right.
And can we use that to our advantage?
I hope we can eventually. Yeah.
Anyway, last one is University of Alaska Fairbanks scientists have discovered and documented the largest known single dinosaur track site in Alaska. This is they're calling it the Denali National Park and Preserve. I thought that was renamed, but maybe the mountain was just renamed. They've dubbed this the coliseum. It's the size of 1.5 football fields, contains layer upon layer of prints of preserved rock. And so what this was an ancient seafloor bed that dinosaurs used to walk across. And when they walked across, they left tracks, and these tracks got fossilized, got hardened. And over years, many, many layers of sediment would drop, and more dinosaurs would walk across, and they'd get more tracks. Then there was, over geological time, a sort of up push in the crust, and this whole seabed ended up turned vertical and sort of sliding apart on a cliff face. Most amazing thing. So when they went, they were like, oh, there's these prints that you should go look at. And the researchers went to look at it, and they said they really were underwhelmed. They found a few prints near the base of this cliff, but other than that, they're like, this isn't really, that big of a deal. They say that as the sun was going down and the light sort of hit it sideways, all of the prints started to pop up. And because it's the seafloor bed with all these layers that had been built up of sediment and new footprints being put in by different generations of hadrosaurs and T-Rexes and what have you, as the thing turns and slid, you're actually looking at different eras, different years, different layers of footprints beneath the footprints as they're sort of sliding out from this cliff face. It's really an amazing sight and is completely just covered in dinosaur tracks.
I still wonder, how do you know it's a dinosaur track and not just a little divot from the wind or the rain or whatever? I mean,
so
impressive.
This was, actually some of it they're saying is so detailed, so well preserved, so detailed that they're like, you can see skin imprint outlines, you can see toes, there's like, some of it has really high levels of detail to it. Also makes you wonder, like, are there other areas of footprints that have footprints hiding below them?
Yeah, if these are all sediments that sit on top of each other, right?
Yeah.
But if a footprint gets filled in, does it still exist as a footprint? I guess it does, because it's from a different
I wonder
layer.
if you could, if deep ground penetration with the right way could find more of these. I don't know how. Then again, I don't know. Like, I get it. Every paleontologist is like be really excited. Like, how many do you really need?
All of them. I want all of the footprints.
You need all of the data all of the time, every time.
What is this? This tells us that these dinosaurs were running around there. What does this tell us about? I mean, is it just, okay, we now see these footprints? I mean, what does it put together for the environment? Is this...
Well, it wasn't just dinosaur prints, too. There's also, like, apparently flora and fauna. I mean, like flora, plant imprints and stuff. So there's actually, this is 70 million years ago, so Alaska was pretty tropical, apparently. It was a very lush, this is a very lush area with all kinds of, the flying dinosaurs and the walking dinosaurs and the carnivorous dinosaurs and even dinosaurs that preferred fungus burgers.
Get your Jurassic fungus burger at Burger for Health.
So it creates a really nice snapshot of everything that was active in the area, along with the plants that they would have been encountering, and it creates the picture of an ecosystem. 70 million years. The planet Earth is like a time capsule.
If we know how to read it, right?
If you know
how to uncover it. Yeah. That's so cool. Mm-hmm. I'd like to go to the Coliseum someday. Denali. I've never been to Denali. That would be an amazing trip. TWIS trip. We need to do it. Let's find dinosaur tracks, everyone. I have a story I want to talk about. The octopus brain. Researchers want to know how octopuses learn and remember things. And in this recent study that was just published in eLife, researchers from Hebrew University in Israel, Harvard University, and also Rupin Academic Center in Israel have been trying to figure out how the nervous system of the octopus works together to allow it to learn and remember stuff. Because we know the octopus is smart, generally. Octopuses as a group of animals, right? So they looked at a very common octopus. It's a common octopus. It's called Octopus vulgaris. That's
the octopus is always telling all those awful jokes.
Yeah, it's vulgar. They checked out a part of its brain called the vertical lobe, and this vertical lobe is known to be involved in behavior related to long-term memory. And so they're like, all right, let's look at these neurons. Let's see how they piece together. And let's figure out what's going on in there. And so they connected a whole bunch of things, and they determined that these octopus brains, this part of the brain, the vertical lobe, is really special in that it manages long-term memory using something called long-term potentiation, which is also found in mammalian brains. However, it does it in a different way. So long-term potentiation in the mammalian brain, we have these particular receptors and we have circuits that are involved in connecting through what's called a Hebbian mechanism of association. And so these associative memories in the mammalian brain happen related to ion flow and receptors that are present in particular areas of the brain. However, the octopus has these really neat vertical circuits. It's beautiful. They've done some beautiful work putting these pictures together of how the neurons connect within the vertical lobe of the octopus brain. And what they determined is that there are basically like 7 different cell types in there. There's a couple of cell types that are called amacrine cells that do the majority of the work. And there are simple amacrine cells and there are complex amacrine cells. And the simple and complex amacrine cells both work on a feed-forward manner. They both go information, 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 and they tell the motor neurons and everything else in the octopus what to do. The simple amacrine cells are more likely to just full speed ahead, just go, go, go. And the complex amacrine cells are like the moderators. They're like, we're still giving you information, but you don't necessarily need to do it as much as you were going to do it before. So they're like modulating the activity. But the entire system ends up recreating long-term potentiation, which is found in mammalian and other vertebrate species, but in a different way, using different mechanisms, doing a different cortical or neural pathway. It's like they figured out a different way to do something that we know we do. And that's the cool part. So now they can compare between the octopuses and the vertebrates and other species that have this long-term potentiation to be able to see if there's lots of big differences. Octopuses have really interesting brains.
Yeah, cool. There's more than one strategy to an active, intelligent brain.
And I think that's the very interesting perspective with this, which is that we know that that of course the active, intelligent brain of an octopus is different. Like they've got different limbs, they've got a whole different setup. They're invertebrates, we're vertebrates. You know, everything's different there. But this is part of that learning for us that there's not just one way to solve the problem, and that when we look at neural networks and the way that information processes through a neural network, there are different ways to put it together to come to a solution that's very similar.
This is also, I think, the greatest evidence for me that none of the eyewitness reports of alien encounters.
Wait, what?
Like, well, if you take those, they usually describe a hominin, right? Usually describe something that's human, basically a human ancestor that would have to be removed by evolution. If you went to the way the evolution finds solutions to things isn't just to say, oh, here's where we're heading, here's the one thing in the billions of stars that we would create as life, and it's not a linear progression. There's other strategies that could be taken advantage of, to the point where if we ever got visited by interstellar
octopuses,
traveling civilizations, they wouldn't be hominins. They would have, They might be, and I think I would be just as surprised if they looked like tardigrades or octopuses, because those are also Earthlings. Those are also Earth solutions.
Right. So different planets are going to have different solutions, and that's
different
part of it.
solutions will have to handling problems, to overcoming things, to build on, to improve on, and once you've started building and improving on a thing, you keep that form. The octopuses are unlikely to go, this whole 8 tentacles is just more complicated than it needs to be. Why don't we just focus on becoming bipedal and using just 2 of them to manipulate objects? Like, it doesn't make any sense. So
yeah. So
this anytime I see
part,
something like this that shows a different strategy to do something that we sort of fundamentally take for granted, as mammalians, like that's just how a brain should work.
A different brain.
Yeah,
and that learning and associative learning specifically is not always the same exact mechanism as of what we have learned for long-term potentiation with NMDA receptors in vertebrate brains, according to
variation
Hebbian
on one planet.
biology. It's
on
just
one
different.
planet we have variation
in one system. Yeah.
Yeah.
Feed forward, though. It's very exciting. Researchers are interested in this because of where it could take us for understanding memory networks generally.
John Hogan says, don't they have separate brains in their tentacles? Well, they've got neuronal tissue elsewhere. So you have, from your colon up to your esophagus, your entire digestive system is lined with neuronal cells.
Absolutely.
Which you could say you have a brain in your stomach, but do you?
It pushes you to do things.
But you have neuronal tissue.
Yeah.
That,
When is a brain a brain? When is a brain not a brain? When is a collection of neurons a brain? These are very interesting questions that should be addressed. So now, of course, let's talk about fly sex.
Of course.
Yeah. So a recent study in Cell has just been released, investigating the neural circuit for sexual behavior in male fruit flies. And in this study, researchers have determined that there is indeed a circuit that they can control. And at the flick of a switch, they can turn male needs or wants on or off. And in addition to that, they can even teach the fruit flies how to push a button with their nose and turn their own needs and wants on or off.
Add your own correlation to modern male human existence.
These are flies. These are not modern male humans. These are just flies, and they're looking at the various areas of the fly brains to see how they were stimulated. Actually, wait, I thought this was flies. No, this is mice. Oh my goodness, I'm totally. I thought it was flies. I've been off circuit for male reward. I was convinced it was fruit flies, but it's mice. I think in my brain I thought they were the same thing, but they're not. These mice in this study, again, still published in Cell, they were trained to understand that if they hit a button with their nose, that they could turn their own needs and wants on and off to promote or not promote mating. And the researchers were also able to turn off the neurons that were related to mating to make it so the males didn't want to mate if they were around females, or so that they were so interested in mating that they would want to mate with inanimate objects.
Oh gosh.
Yes.
So now I want to figure out.
It's not flies. It's mice. Now I want to know if we could do this in flies. I'm sure.
Right? We could do it in flies. Now so what's really interesting is that you think of especially sexual drive as largely deriving from sort of an automated maybe hormonal and conceptual to some degree. Part of the brain that you have control, but has desire and impulse, a thing like this, where you have in mice where they would be attracted to, or wanting to reproduce. So if you've turned off that signaling, but you've given the mouse the option to hit a button to turn it back on, what is that the drive? What is that separate from already being told by this sort of what would you call it? Is it not executive function, but this sort of regulatory functions of the brain that are telling
hormones that are involved in sex drive and how that works. Yeah.
Now you've removed those impulses. But the mouse can hit the button and decide, I would like to switch that. Or the mouse can be like, oh yeah, this is not the right time to be feeling like this. I got to hit that button and turn it off.
Yeah, so they can figure that out. These researchers went into an area of the mouse brain that they call BNST, and they started putting something that's called substance P, and this is in the hypothalamus, and the substance P seemed to be very important for being secreted from these neurons, and they accelerated activity and ramped things up. Not only the drive, but also it impacted what's called the refractory period. So normally after a mating period, males have a refractory period of a stretch of time. And this research that they published was able to shorten the refractory period so that the males, instead of having like 10 to 15 minutes or 90-minute delay, that the males wanted to mate once again and were driven to mate once again. So they had what they quote as more than 400,000-fold reduction in the refractory period. Make the males more ready. Substance P containing neurons in the preoptic hypothalamus go down to brain centers that are critical to movement and possibly anticipating or experiencing pleasure. And this could be very important for understanding hyperactive sex drives, lack of sex drive, being able to discover ways to boost sex drive or decrease it therapeutically. Maybe there are molecules or drugs that could be developed following up on this.
Nice.
I can't believe I totally read this study and I thought it was flies. I think I was reading about the dead flies and then I thought
conflated.
I conflated the flies and I'm very sorry, everybody. It's not sexy flies. It's sexy mice.
Everybody forgave you a long time ago. They actually forgot, and you just reminded. Oh, she did make that mistake. I forgot about it. Gosh darn it. I was thinking flies, and it turned out to be mice. Now I want my money back. Oh, good thing the show is free.
That's right. Have we done it? Have we made it to the end of our show tonight?
I think we might be there. Yeah.
I think we might have done all the stories we were going to do. Thank you, everyone who's been part of the chat. Thank you, everyone who is in the Discord. Thank you, Fada, for your help with the creating of the social medias and the show notes. Thank you, Identity4, for recording the show. Thank you, Gord, Aaron, Lore, others who help keep the chat rooms very nice, happy, safe places to be. Thank you, Rachel, for editing the show. And as always, thank you so much to our Patreon sponsors who are allowing us to really make this show possible. Thank you to Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kenton Northcote, Rick Loveman, George Corus, B.R. Velasarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Sarah Forfar, Don Munda, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredos 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leesman, a.k.a. Z, McKen Hayes, Howard Tan, Christopher Rappin, Richard, Brendan, Minnis, Johnny Gridley, Jimmy, Day, G. Burton, Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, you can find information at twis.org. Just click on that Patreon button. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, and again, 5 a.m. Thursday, Central European time.
Same time.
Broadcasting live from our YouTube and Facebook channels. And from twis.org/live. And from Twitch.
I gotta change that. Okay. Oh, want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, please get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. What's our website? www.twis.org. And you can also sign up for a newsletter. We won't send you one, but you can sign up for one.
We might send you one. It's a thing. We've done it before. You can also contact us directly. Email me, Kirsten at thisweekinscience.com. Justin is at twistminion@gmail.com. Blair is at blairbaz@twis.org. And if you want to make sure that it doesn't get spam filtered into oblivion, put TWIS into that subject line, especially if you want Blair to get your congratulations for the new baby in her family. Yes.
You can also hit us up on, are we doing the what do you call it, the platform formerly known as Twitter? Are we still? I don't think I've been there for a while. If you go there, though, you can find at TWIS Science, at Dr. Kiki, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
And we will be back here again next week. We hope you'll join us again for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may...
Or you just might understand, or you might not understand. This is the after show. Once again, I do not know where Justin has gone. He has family considerations over there. It is work time. Harry, there he is. Hello. There's Justin, little baby.
Where there?
Look at that. Oh, there. Hello. Hello, little one. This is what little ones look like when they are a little older than what Blair's little one is looking like right now.
They start out very small.
Small and little squirmies and squishy faces and all the cutenesses. Yes. We normally do after showy type stuff, but oh, look at that, headphones. There's a born podcaster. He wants to eat it. Don't eat the headphones. You want to eat the headphones? Who's there? Can you hear me? Hello. Hey. Hi, hi. Hey, hey. Yeah. You like. Yes, we're just all the TWIS babies right now. At some point, we'll bring the TWIS babies together. Yeah, we normally do after show, but I see you have child duties, so I don't want to push it a lot further. So
okay. Well, I will see you next week.
Yeah. I love seeing the little one. Great to do a show with you again this week, Justin. We all wish Blair well and hope that she's
can't wait to meet the Are we allowed to say that it's a human baby?
It's a human baby.
Okay.
Yes. Oh, chocolate. Oh, here, Daddy. Have a cold that I caught at my little school.
Oh,
I love you. Now be sick, okay? Yeah. Little germies. They're wonderful. All right. Say good morning, Justin.
Good morning, Justin. Say good night, Kiki.
Good night, Kiki. Good night, everyone. Thank you so much for joining us for another episode of This Week in Science. We'll be back again next week, 8 p.m. Pacific time on Wednesday. Stay safe, stay curious, and stay lucky.